There it is, all good. Okay, thanks Natalia for your introduction, and thanks to Neda for the great overview we had about the world of imaging genetics and its relevance for Alzheimer's disease. And in my talk, and I think also in the next talks, we're going to try to zoom in a little bit and see how we can use these methods to understand the alterations that we see in imaging endophenotypes in the preclinical and clinical stages of Alzheimer's disease. And specifically today, I'm going to show this work where we try to use polygenic information to characterize the biological process, the biological pathways that determine multimodal, so alteration from different MRI sequences. So you've seen this picture already in Neda's presentation. We know that Alzheimer's disease is a complex interplay between genetic and environmental factors, and some types of Alzheimer's disease, like the familiar one, are strongly determined by rare variants like the APP or the PSEN1 or 2 gene, but also the most common late onset Alzheimer's disease have a strong genetic influence. We all know about the ApoE4 gene, which has a strong influence, and then having an E4 allele strongly impact your risk of getting Alzheimer's disease. But recent genome-wide association studies, GWAS, have shown that there might be a lot of different single nucleotide polymorphisms, NIPs, so genetic variants, that seems to increase your risk of getting Alzheimer's disease. And while, as expected, most of these NIPs are related to biological processes that are within the context of amyloid deposition, amyloid production, or clearance, as amyloid being the bed proteins of Alzheimer's disease, functional enrichment studies have shown that there might be a lot of other different biological processes that are encoded within this polygenic risk of Alzheimer's. For example, lipid metabolism or endocytosis clearance pathways, but also neuroinflammation, immune activation pathways, as well as synaptic plasticity and function. And this genetic heterogeneity does also end up in the heterogeneity in the imaging phenotypes that we observe throughout the stages of Alzheimer's disease, and especially in the early stages. Indeed, multiple imaging studies using, for example, amyloid or tau PETs, but also structural, functional, and diffusion MRI have shown different patterns of alteration at the individual level from the early stages of Alzheimer's disease. So while these markers give us an information about the alterations in the brain, we don't know what are the biological processes that underlie it, that cause these alterations. And so it is important to link the genetic level to the imaging level to understand this. So the aim of this study was exactly this. So we wanted to biologically characterize altered image-derived phenotypes in Alzheimer's disease, and specifically early Alzheimer's disease, and investigate the role of polygenic risk scores and the associated pathways in determining this alteration in cognitively unimpaired individuals. Just briefly, to do that, we use a cohort called European Prevention of Alzheimer's Dementia cohort. It's the European cohort targeting the early stages of the disease and including people with age above 50 and no diagnosis of dementia, so with a clinical dementia rating score below 1. And try to characterize them in many ways using, for example, cerebrospinal fluid data, but also in some cases amyloid PET data. And of course, for this study, we also had genetic data and imaging data. And for imaging data, we're going to focus on structural, so T1 and FLAIR, but also on more advanced data such as resting state fMRI and DTI, diffusion-weighted images. Just to give you an overview of the cohort, these are the numbers. So overall, we had 1,700 participants, and this is also stratified by 80 stages, which means whether they had amyloid or tau. But I think just to highlight a couple of things, they had my age around 65, and you can see the MMSE was around 28, and the clinical dementia rating score only 30% at 0.5, which is a very mild cognitive impairment. So again, we're looking at non-demented individuals and trying to characterize the early stages of the disease. So for genetics, what we did was to look at the most recent GWAS for Alzheimer's disease, which found out that there were 85 SNPs that are related to Alzheimer's dementia. And then we had our data from EPAD, and we computed polygenic risk scores. So we computed scores that told us how much was the risk of each individual to get Alzheimer's disease, but not only based on one gene, but based on 85 SNPs. And we compute two global polygenic risk scores, one including ApoE, which is the strongest determinant, but also one without it, because we wanted to look at the effect beyond ApoE. But we also wanted to go for pathways, so we did a pathway analysis. And how we did that was to map each of these 85 SNPs to specific genes, and then look for, do something called gene set enrichment analysis. So look for what are the biological pathways that are enriched, that are strongly expressed within this set of genes. And this is done through a survey which is called gene ontology. So we got a very long list of biological pathways that are encoded within these genes, and then we did a clustering analysis to see whether there were some clusters within, some cluster structure within this huge list. And here I show the results of this analysis. We found six pathways, six clusters that were enriched within the risk of Alzheimer's disease. We labeled them based on the number of times a word would appear within the description of this pathway. And as you can see, we call them immune activation, signal transduction pathway, inflammatory pathways, and then we had the migration, amyloid, and clearance pathway. And I think what is interesting here is that you can see these are also split in two big clusters. So the first three are more related to inflammatory process in general or immune activation process, while the second three seems to be more Alzheimer's specific. So we have amyloid there, which is the biggest, of course, but we also have pathways related to cholesterol, lipid, or clearance. And this is not very easy to read, but this is just the contribution of each gene to each of these pathways. And just to highlight that some of them, like the one that you see all the way to the right, are very specific. So for example, TREM-L2 was very specifically contributing to the immune activation pathways, but other genes are more spread around all these pathways. So we wanted to predict some outcomes with this genetic information, the global PRS and the pathway PRS. And in terms of outcome, we had CSF markers, but today we're going to focus on the imaging ones. And for this, we used the radiological assessment of images, especially related to cerebrovascular disease, and a series of multimodal quantitative MRI figures. All of these I'm going to explain in the next slides. But for each of the outcome, with run simple linear models investigating the effect of our two global polygenic risk scores, but also of the six pathways on these markers, and did a series of corrections, as is usual in these studies, to control for multiple comparison. So starting with the radiological features of cerebrovascular disease, here we focus on something called the FASICA scale. This is a scale that evaluates the presence of these white spots in your white matters. It's a scale that goes from 0 to 3, with 0 not having any white matter lesion and 3 having a lot of them. We also looked at the presence or absence of microbleeds, so small hemorrhages in your brain, and we divided participants as having less than 2 or more than 2, as based on the most recent criteria. And eventually, we look at enlarged perivascular spaces, which are these small spaces within the perivascular space. And this is a 0 to 4 scale, again, with 0 having no enlarged perivascular spaces and 4 having a lot of them. And when we look at the relationship of the pathways and the PRS with these markers, this is a very confusing figure, but I can focus your attention on some of them. What we found was that mostly the first 3 pathways, so the one that were on the inflammatory side of things, so the immune activation, you can see the signal transduction a lot, but also the inflammatory pathways were determining a higher scores in all these markers. So meaning that inflammatory pathways might regulate the presence of cerebral small vessel disease radiological features in this case. And now, moving to the quantitative MRI, we started to look at structural MRI-derived phenotypes, and we started from T1 sequence and FLAIR sequence. From T1, we computed hippocampal volume, so we only wanted to focus on Alzheimer's disease-relevant markers, and we used the free surfer pipeline that most of you know to compute hippocampal volumes. And from FLAIR, we use a pipeline called Bayesian model selection to segment white matter hyperintensities and obtain volumes of white matter hyperintensities in specific regions of the brain. Specifically, we parcellated the brain in lobes, so the 4 lobes, but also layers, meaning the distance from the ventricles, which seems to matter when we look at white matter hyperintensities. And so we ended up with 8 regions of interest for our white matter hyperintensities. And when we look at the association with the pathways, here you see the results in this heat map. In the hippocampal volumes on the bottom row, we only found very mild association, which actually didn't stand after multiple correction, but this was with the migration and with the clearance pathways. But when looking at white matter hyperintensities, this was very specific, associated with what we call the clearance pathway. So a pathway which seems to be more related to endocytosis, lymphatic system in general. It might also be important for white matter health. Then we move to more advanced sequences. So we look at diffusion imaging, and we use DTI, so TBSS pipeline, to compute DTI scalars. And we computed fractional anisotropy and mean diffusivity in 10 regions of interest. These regions have been previously shown to be altered in Alzheimer's disease in many ways, and we grouped them in commissural fibers, limbic fibers, associative fibers, and projectural fibers, as you can see in this slide. And again, when we looked at the association with the pathways, again, we have the heat map here. You can see there is an initial association with the global PRS, which is the first column, but we found a very specific association, this time with another pathway, which was called we call the migration pathway. This one is more related to phospholipid metabolism, cholesterol production, or reflux, so it seems to also make sense in terms of white matter integrity. And finally, our favorite, we move to the functional MRI, and in this case, we use a dual regression approach to extract within resting state network connectivity. But we only wanted to look again at Alzheimer's disease specific markers, so we couldn't focus on all the possible networks. We only looked at it within 3 sub-portions of the default mode network, which has been shown in many studies to be altered in many different phases of Alzheimer's disease. And again, in these smaller heat maps, this time you can see the results. They're a little bit less, but very interestingly, again, we found that only in this case, the signal transduction pathways was determining alterations or changes in the ventral DMN, which is also one of the earliest alterations in Alzheimer's disease. And in this case, this pathway was in the inflammatory side of it, but in this pathway, you also see a lot of genes coding for synaptic functioning, synaptic plasticity, so it makes sense that it relates to the functional alterations in the brain. So with this, I'm going to conclude, and first of all, from the Alzheimer's perspective, I want to say it was already interesting to see that within the polygenic risk of Alzheimer's, we could find 2 main families of pathways, one that seems to be Alzheimer's specific, so with amyloid, clearance, and the lipid metabolism, but one might be non-specific, and it's growing a lot of the evidence about the role of inflammatory process in many different neurodegenerative diseases. So this could be a non-specific factor to many different diseases. But from the imaging side, I think it was very interesting to observe that we found very specific association of specific biological process determining distinct imaging markers. So we might disentangle what are the processes determining this. So overall, we think that polygenic pathways, we show that polygenic pathways differentially promote distinct neuropathological features and enhance the disease heterogeneity that we see from the early stages, and the end in Alzheimer's disease through different alterations, through different pathways. So with this, I would like to thank you for your attention. I would like to thank all my collaborators for helping me, and Natalia and Tavia for organizing this very nice symposium.